Meiosis starts after an interphase, which is not different that of intermitotic division. During the pre-meiotic DNA duplication has occurred at the S phase of interphase. Apparently, there is a decisive change that directs the cell towards the meiosis. Further, in the beginning of the first meiotic division, nucleus of the meiosis starts to swell up by absorbing the water from the cytoplasm, and the nucleus volume increases about three folds. After these changes, the nucleus first meiotic division of first phase, that which is known as prophase. First prophase of the meiotic division is the longest phase. It includes following five substages. It includes following six substages. Proleptonema Proleptotin is closely resembled with the Eldry mitotic prophase. Eldry mitotic prophase. It resembles the Eldry mitotic prophase. Now, Eldry mitotic prophase, how will be? This stage, chromosomes are long, thin, and uncoiled. Embryos duplicates and moving opposite poles of the nucleus. Longitudinally, they are single and thread-like structures. In leptotin stage, chromosomes are more uncoiled and more thread-like structures. Thread-like structures. So means like this. Long, uncoiled and thread-like structures. In this stage, chromosomes take a specific orientation in the nucleus. The ends of the chromosomes converge towards to one side of the nucleus. They are converged. These two are converged. Two ends of the nucleus converge one side of the nucleus. So, like this, they are converged. That side where centrosome lies. Ends of the nucleus converge one side of the nucleus. So, like this, they are converged. That side where centrosome lies. The centrioles duplicates moving towards the opposite pole of the nucleus, and thus each pole of the cell possesses two centrioles of the single diplose, as of now, zygote. The zygote pairing of homologous chromosomes takes place. Homologous chromosomes come from mother ova and another come from sperm, attract each other, which are pairing start. The pairing of the homologous chromosomes is known as synapsis. Of the homologous chromosomes is known as synapsis. Synapsis points along the homologous chromosomes. The pairing of the chromosomes is known as synapsis. So there are the many points along the length of the homologous chromosomes. homologous chromosomes come from mother ova. There are three types of the synapses have been recognized. Firstly, pro-terminal synapses means the pairing starts from the terminal towards the center. This type of synapses is known as pro-terminal synapse. Second one is procentric synapse. They start the pairing from the center and moves towards the end. So this is called as procentric synapse. It is known as procentric synapse. They move towards the end. Type is localized synapse or pairing. Localized pairing. Or random synapse or random synapse. In this type, synapse may occur any point along the chromosome at the center, end or anywhere. So that is called as random synapse or localized pairing. Pairing of homologous chromosomes are exact and specific. Alignment of the chromosomes exactly gene for gene. The paired homologous chromosomes joined by 0.2 millimicron thick proteinaceous synaptal complex. 0.2 millimicrons of the synaptal complex. This complex along the whole length of chromosomes. Usually, they are anchored with the nuclear envelope. Synaptal complex stabilize the pairing of homologous chromosomes. They stabilize the pairing of the homologous chromosome. Otherwise, they are divert one to another. No, it's called recombination or crossing over. Recombination and crossing over. So here what will be? Both the chromosomes are exchanged in this stage. Like this, they are exchanging. So these are the homologous chromosomes. These two are called as diads. These are called tetrad. These two are called homologous chromosomes. So after this, what will be? So another chromosome. If you see the different color I am showing. So these are they are exchanging recombinase. Recombinase, one of the enzyme. So facilitate this recombination means these exchanges of the genetic material occur. Exchanges of the genetic material. So these two are homologous. These are heterochromosome means these are the sister chromatids. These two are sister chromatids. These two are sister chromatids. But between these recombinase, one of the enzyme facilitate the exchange of the crossing over. So in this way, the crossing over occur and facilitate cytogenetical activity. In which organism synaptal complex won't be occur this crossing over. In example, Drosophila melanogaster. Actually, doubling of the chromosomes would have occurred before the earlier prophase. So, generally, it would happen before the duplication of the DNA. So in pactin we can be seen in this very clearly. In pactin, what would happen? They are twisted. They in these non-sister chromatins become twisted each other. They are twisted each other, cannot distinguish separately. They are twisted each other, cannot be distinguished separately, we can observe. In the middle of the packeting stage, they splits longitudinally, form a two chromatids. Two chromatids formation takes place longitudinally. Transversely also, if you take the transversely, four chromatids. Four chromatids look like means dyad and tetrad. Like that, it will be however through the earlier part of the mitotic prophase, chromosomes behave as a single body. Is now changed. In this stage, in the packeting stage, the chromatid of the chromosome behave as a half of the DNA. Now they are behaving as a independent of each other. Although still they continue to be linked together by its common centromere. Each synaptal pair, commonly referred to as a bivalents or dyads, they are because they are two visible bivalents, two visible chromatids are there. That's why it is these are called as dyads. As well as they are at this point they are called as quadrivalent or chromatid, four chromatids. So because they are four visible chromatids. During the packeting stage, the important phenomenon is called as crossing over. At this point, this is called chiasma. So this process and phenomena is called crossing over because they are exchanged the their genetic material and variation occur. Variation means new characters appear. They are neither like mother nor father. Crossing over involves the reshuffling, redistribution, and mutual exchange of the genetic material of their parents between these homologous chromosomes. This is one homologous chromosome, this is one homologous chromosome. Both are now considered as a heterogeneous chromosomes. But they are the genetic material exchange not between these two. Only one of this, one of this, or one of this, one of this. Like this they exchange. Not between these, not between these. Keep remember this. According to recent views, the chromatids of the chromosomes they are divided transversely and with the help of the endonuclease. Endonuclease. With the help of endonuclease, they divide and look like a four chromatids of the homologous chromosome. One homologous chromosome only transversely they divide with the help of endonuclease looks like a four chromatids, which is reported to increase the nucleus. Means doubling of the DNA will be no the amount of DNA that the same chromosome same nucleus will be, but amount only doubling. Amount of DNA only doubling, not this. During this stage, by Sterna and Hota, Sterna, Sterna and Hota, both scientists in 1969, they are reported. Both are reported. In this way happens. After division of chromatids, The interchange of the chromatids takes place and between non-sister chromosomes of homologous chromosomes. The broken chromatids are united with the help of the ligase, with the help of an enzyme ligase. What is this? Ligase. So this is like this, they exchange. The process of inter-exchange of the genetic material between the chromatids of homologous non-sister chromatids is known as crossing over. This phenomenon is called crossing over, accompanied with the formation of chiasmata. Formation of chiasmata. So this process is called a crossing over, where these points are attached. This point is known as chiasmata. Chiasmata formation takes place where they are attached. That point only. Putta and Sternum both are in 1969 reported that in packaging stage and zygote stage, both stages DNA, small amount of the DNA formation takes place. Both stages. The amount of DNA you used wherever they are during this stage, they used to utilize it for the broken DNA during the crossing over and chiasmata. Wherever these are uh, cut place, no DNA cut places will be. So here and there these this DNA which is increased during the pactin and zygote, they are utilized in this stage during the pactin and zygote is prominent to be up to this stage, and it is remains in the nucleolar organizer. Nucleolus means center of the nucleus, no? At the center, the nucleus, the dark strain structure that is known as nucleus, no? Up to this stage, they are very prominent. It is found to be associated with the nucleolar organizer. Nucleolar organizer. So, diplotin. In this stage, desynapsis. The desynapsis takes place, means unpairing of the chromosomes occurs. They are separating. In this stage, chiasmata first seen. In this stage, these chromatids are clearly visible, but synaptal complex appears to be disappear. These are appear to be disappear, means gradually disappearing. Leaving the participating of the chromatids of the homologous chromosomes to places discrete points is known as chiasmata. Cross piece, like this they are attached, like this. So this is cross piece. Often there is unfolding of the chromosomes, so allowing the RNA synthesis and cellular growth, RNA synthesis and cellular growth. Wherever they are chiasmata and all, they are allowing the RNA and cellular growth both, including the proleptonema. So this is becoming the sixth stage of this meiotic division, that is diakinesis. In this diakinesis, so the chromosomes are more condensed, evenly distributed into the nucleus. So all over the nucleus they are evenly distributed. So like this, they are condensed, thickened and so are evenly distributed in this diakinesis.